that picture and tell me what you see. Some of you might have seen this before. What do you see? Okay, who wants to raise a hand and tell me? What do you see? You see a woman, an old woman or a young woman? Do you see an old woman or a young woman? Who sees an old woman? Who sees a young woman? So the name of this picture is My Wife, My Mother-in-Law. Okay? And it has two different people in it, depending on what you look. So the old woman, this is her nose, that's her eye, this is her hair, and this is her shawl, and that's her mouth. That's the old woman. The young woman is kind of looking away. This is her nose and her eyebrow, and that's her chin, ear. This is her hair, and she has a fancy hat on. So you could see either my mother or my mother-in-law. Okay, this was made about 100 years ago, 1915. And the point is you can't see both at once. You could see one or the other. But when you have that input going to your eyes, it's the same either way, right? The sensation, black and white dots hitting your retina, is the same whether you see the mother or the mother-in-law. What's changing is your perception, how your brain has processed that, right? It's either you'll see the young person or the older person. So what happens is this input goes into your brain and it's comparing it with something in your brain, either an image of a young woman or an image of an older woman. And part of what you're going to be doing this semester is figuring out how the brain works. And we want to start out right now showing you the difference between sensation, what's coming into your brain, and perception, what you make of that information. This was actually the original picture. This was from 1888 postcard, German postcard, my mother and my mother-in-law. And here's the eye of the mother-in-law, her nose, mouth. And for the young woman, you could see her, that's her little nose right there. She's kind of turning away, going there, on it, right? The original image a long time ago. So this has been going on for a while. So what you're going to be doing this semester is figuring out the brain, okay? So now I've got you interested in how cool a system this is. That's not going to be very straightforward. I'd like to introduce our TAs. All right, you're going to be working with them very closely. Maybe you can all stand up. Our head TA is Yushi. She's going to be the one who's going to kind of run a lot of the things here and help you. We have 3 of our own students helping out, and actually Allie, Miguel, and Andy, maybe just introduce yourselves real quick, and you too, Yuji, as well. You start.
Okay. Hi everyone, I'm Yuchi Guo, and I'm a second-year PhD student in Dr. Wang's lab, and I'm the head TA of the 4.
Allie?
I'm Allie Burgess. I'm a senior BME, and I'm looking forward to TAing.
I'm Miguel, senior BME. Hopefully you recognize me from last semester.
Hopefully you do, guys, okay?
I'm Andy. I'm also one of your TAs. I'm also a senior BME.
Okay, great, guys. So you're going to be seeing a lot of them this semester as well, okay? So what exactly does the brain do, all right? You start by getting some stimuli in from the outside world, right? Some environmental energy, and this can be light, different colors, which hit your retina, or the picture, which I just showed you. Sound, you'll hear all kinds of different frequencies, which could be just a lovely song, or it could be discordant yelling. Mechanical stimulation, temperature, pain, there's pricking pain and burning pain, vibrations. You could tell if a train is running by right next to this room. There's all kinds of receptors in your fingertips especially that can tell you all kinds of things. In this particular case, a drum is rolling around, and you could feel whether or not there's anything on that, smooth versus rough. Hot versus cold. We have different kinds of receptors for that. Chemical, olfaction, smelling, and this is another thing where sensation and perception could be different. You could smell something and think it's wonderful, your mother's cooking or whatever, and depending on maybe your culture or ethnic background, that same smell might be not so nice at all for somebody else. So how you perceive things could be very different. So after you get this stimuli coming in, these peripheral receptors are going to respond to it. They're going to fire at some very specific rate, depending on what that is. So at a certain temperature, you'll get a certain firing rate in these neurons. Guys, I'm just giving you a general overview today, so you don't have to take any notes, because you're going to get all this again. So for example, if you had the letter A, and you were rubbing your finger over the letter A, right? You have all these little receptors in your finger, and at some little points, all these little receptors are the white dots here. And wherever you have that letter A sticking up, you can, by recording from peripheral nerves going towards your brain, record when that's on or off, or you can do the same for the letter B and C, and actually come up with something in those nerves, firing rates, that look like the letters you just had running over those receptors. This is an example of a kind of receptor zone that responds to maybe that letter A being popped up, and it's going to result in some neuroactivity, based on how big that is and everything else. So then we have these ascending pathways. So this information maybe is in our finger, right? But we know in our brain we've touched something. So how does it get up to our brain? Well, we have these little receptors, maybe, and they come all the way. This nerve comes in with information all the way to the spinal cord, and then it travels. And that's a long nerve, by the way. There's no break there. So you can have nerves that are like 1 meter long. Comes in, it'll synapse in the spinal cord, and they go all the way up to the brain. So we have this pathway heading up through these ascending pathways, and then we have synaptic zones. So you have a receptor turned on here, okay? And it's going to cause a nerve to fire. And now what happens is you can have convergence, lots of neurons coming together and firing on the next nerve. You don't also have divergence, in which a nerve comes on and fires on lots of different things. So you have these synaptic zones where you're sending the information maybe to more than one place, or information is coming in from lots of different places and converging together. And then you're going to go to the brain. And your brain is so super organized. You're going to spend a lot of this semester learning about that. But this whole back part of your brain here has a lot of different, very defined areas that respond only to auditory stimuli, or only to visual stimuli, or only to touching something on a table. And you could tell if this is soft or hard or hot or cold. Extremely organized places in your brain. So it's not like you come up these ascending pathways and just go anywhere. It goes to a very specific location depending on what kind of receptor was activated and where it was activated. So you touch something with this finger, it's going to go to a different place in the brain than if you touch something with your toe. Very organized, okay? So you have these cortical maps, and then you have an association with your memory, just like you just did with that picture, right? You either associated those black and white images with a young lady or an old lady. If you hear a dog, you have an image of a dog in your head. You hear a baby crying, you have an image of a baby crying. So you have these memories that you associate things with. The first time you hear something, you don't have that to compare it with, right? So this is going to affect what you're going to do after that. So after you have this association, you're going to come up with some desired actions. You hear a baby crying, you might want to go run towards it, right? Or do something like that, which will lead to motor commands. How am I going to move to respond to either touching a hot stove or whatever it is? And so our motor area is another part of the brain, okay? This front part here is going to send signals down to our muscles and say, hey, I want you to move this arm or this finger or whatever it is. And obviously, in order for that to take place, you have to have a lot of talking going on between what happened here and what you perceived, right, your perception, and then what you want to do about it here. So let's say you grab a hot pot off a stove, right, and the handle's super hot. Have you ever done that? And your initial thing is to let go and drop it. But if there's like a little toddler right there, you're like, no, I'm not going to do that. I'd rather burn. So you get to decide kind of what you want to do with a lot of these things, right? More synaptic zones. So you have this plan coming down from your brain, and do you only need to turn on a couple of muscles in a finger, or does your whole arm need to move out of the way of something, right? All this inner talking between neurons can take place. So now you have some descending pathways. So you have this motor area of your brain going to send a signal down to your spinal cord, and then from there we're going to go to local spinal circuits within your spinal cord, and within your spinal cord you're going to activate a motor neuron, which will go to the muscle, and then you'll get your muscles to contract, and this part you know from last semester, right, the whole actin myosin thing, and the result is movement, right? So we have this really long, complicated almost process of how we respond to different things and what our brain does to decide interactions. And a lot of what you're going to be doing this semester is figuring out what is this that's taking place. Once you have information coming in, we actually know a lot about how stimuli activate peripheral receptors and lead to firing of nerves, and then how that gets up to the brain. And then there's a lot of, we're not really sure how that can lead to motor commands and actions. And part of why this is relevant is if you think of people with diseases. That's how we figure out a lot of how the brain works, right? If somebody has a stroke, one side of their brain was damaged and they can't move the other side, and that's a little obvious one. But what about someone with Parkinson's? their muscles work, but they're not working right? They have a very jerky movement. Or autism, they can hear, but they're not responding. So some people's brains aren't having the same connections, and what you guys are going to be doing this semester is doing a lot of modeling, trying to figure out this whole big area here in this gray box. We know this part actually pretty well, and in fact, for those of you taking System Bioengineering Lab, this is what we're doing in lab this week. Kind of you're going to control muscle movement, get nerves to fire, record the firing, and look at the relationship between the two. So this part we know pretty well, this part we know pretty well, but this whole part in the middle is kind of a little fuzzier. So what you're going to be doing this semester is hearing from 4 different people who are kind of experts in this. Dr. Wang, Dr. Winslow, Dr. Shadmere, and Dr. Zhang. And something you'll notice about all of their topics is that they're computational, right? Computational neuroscience, computational modeling. You're going to apply the math to how the brain works. Taking all these inputs in, coming up with the synaptic zones, and then what's going to happen after that. So that's what you're going to be doing this semester. You have lots of guest lectures. These are experts, people who are doing research in very specific areas of the brain, and they're going to come in. For example, Dr. Connor does research on the visual system. He's going to talk to you in a little more detail about how you process visual information. TAs, we have an email for the TAs, SBE2, period 2017, tas@gmail.com. I will post all these, and I guess we have a link on Blackboard too, so you could email them then. It's easier than emailing them all individually because they'll take turns monitoring this. Each TA has a section on Thursday, and you can see what time they are, and you can email them if you have questions about your section. Exam and grading policy, you'll have 3 exams. Okay, exams 1 and 2 are going to each be 25%, exam 3 is 35%. They're going to be in the evenings, just because we want to give you more than 50 minutes to work on them, okay? And you'll have homeworks due pretty much every week. They're going to count about 15% of your grade. There's 10 of them. And this is a whole big thing about disabilities. I should have gotten an email from the Office of Disability if you get more time in an exam or something. Makeup exams, please let us know as soon as possible if you're going to be out of town or if there's a conflict so that we can arrange it. And then this is a big thing, especially for Dr. Wang. Anyone cheating receives an F in the course and will be reported to the Dean of Students' office. So we want you to work together. I'm going to emphasize that again on the homework. But my degree's from Johns Hopkins, Dr. Wang's degree's from Johns Hopkins. We kind of take it personally if people do anything to ruin the reputation of Johns Hopkins. So we actually do really report immediately to the Dean's office if there's any question about that. So homework policy. Want you guys to work in teams, and what I suggest you do is look at the homework, work on the homework, get together as a group and talk about the homework, put it on the board or a whiteboard in the design studio or whatever it is, work through it all, leave it all there, and go back and do it again your own. And then we don't have to worry about any cheating or copying or anything like that. If there's line for line code copied, that's a violation, okay? Or if we could tell that homework's been copied. If the graders ask us, hey, I think these 2 people worked together and you did, then it's probably too close. But if you all work together and then leave and then do it again on your own, it'll be fine, okay? So please work in groups and arrange groups that work with your schedules, because that's really helpful. Homework is due on Fridays at noon. Okay, gonna upload it to Blackboard as usual. If you turn it in between 12:01 and 1, you get 50% off. After 1 p.m. it's not accepted. Class is at 1:30, and Dr. Wang didn't want people working on homework during class. Okay, so it's kind of due then at the last minute, all right? And academic integrity, again, we're really strict on this, okay? So get help on the code and work together on the general problem, but you should all be doing your own work. You can see a lot of the emphasis in this course is computational modeling of neural systems, so you need to learn how to computationally model. So you need to learn how to do this on your own. It's kind of a big goal of the course. We can't test some of that in a test, right? I can't ask you to do a MATLAB program in a test. So it's all going to be in the homework problems, but it's something we really want everyone here to be able to do well. All right, so I know you don't have clickers with you, but figure out the answer to this. All the following are true except there'll be 3 exams worth 25, 25, and 35%. Homework's due on Fridays at noon is worth 15%. Homework can be submitted late for half credit until 5. There's 4 core lectures and numerous guest lectures, and you should work in groups on homework. Think about it for a second, talk to a neighbor. All right, C, okay? So get that in by 1, okay? All right, so our goal is to make this class one of the most enjoyable you've taken at Hopkins. And actually, based on student reviews, it's a lot of work, but students think it is worth it. By the end of this course, you're going to know something really cool, how the brain works. I mean, does it get any cooler than that, you know? So before class, you should know the topic. This is the visual system, the auditory system. Get a little background information. Come to class, ask questions. Okay, we will be recording the lectures, but it's usually better, I find with students, either hand take notes, don't type anything into a computer, because just summarizing someone's sentences into a couple words is actually a really good learning tool, or just sit and pay attention and get the big gist of it and then go back on with the lectures afterwards. And that's where students seem to get the most out of it. And then after class, let us know if you have any questions on any of the topics. You know you've learned if you can get the homework done and pass the exams, okay? As always, we'll curve this class at the end, try to treat you guys fairly. If you ever have any questions, feel free to email me or any of the TAs or Dr. Wang, okay? I'm not giving any of the lectures, but I know all of you and I'd be happy to sit and meet with you about the class, all right? We have a Facebook page. We started this last year. It's JJU BME 2017? Double I, oh, SB2, okay. So it's here, and we'll post this so you know the address. So last year, about half the students use this, and they use it a lot. They post a lot of stuff. They post stuff during class, and we try to keep up with it and answer questions right away. Some people aren't into it as much, and that's fine, but this is just another way. A lot of you have Facebook very easily accessible, and while you're watching a lecture, can just add a question if that works out. And you're posting the lectures on YouTube? Okay, and there's the link for that, and we'll post this on Blackboard too. So you'll be able to get a hold of that, all right? This is all the things you're going to cover, which I know you can't read. I just want to focus on the first 2 weeks. In the first 2 weeks, you'll notice the word introduction over and over. We're giving you an overview of everything you're going to cover this semester in the next 2 weeks. So starting with me, just the general course, Dr. Schramm's going to start in a few minutes on neuroanatomy, okay? You learned in molecules and cells the importance of structure to function. So there's nerves that are a meter, there's nerves that are small and just connect to everything. Their shape has a lot to do with things, and what connects to what is really important for understanding how it works. Introduction to the Motor System by Reza Shabnir. Okay, we just quickly went through this. Neuroengineering by Dr. Sarma, and then sensory system, which we just again did a very quick example of. And Dr. Zhang is going to give you an introduction to computational neuroscience, how to apply the numbers to how the brain works and model it. And that's your first assignment, actually, is going to be Dr. Zhang's. Okay? So the objectives of this course: understand the general structure and functions of the nervous system and the brain. Understand the basic principles of the sensory systems, which includes auditory and visual and motor processing. And understand the basic principles of neural computations. And when you ask the difference between a typical neuroscience course and a neuroscience course for BME students, this is where this really comes into play. Okay, you guys are going to be able to apply math and programming skills to understanding how this works. All right. The textbook for the class is Purvey's, and it's kind of the way you had from SBU1. It's great to have some kind of textbook to get some background reading, but there's no specific required readings for each time. If you want to share a textbook with two of you or something, that's probably fine. This actually, though, I think is a pretty good book and does a nice job. There's specific chapters, and again, this will all be posted, that are pretty relevant to what you'll be discussing this semester. Okay? There's other books that that was a neuroscience book, Purvey's, and which you might take in a neuroscience class. There's some that are more computationally oriented that some of you might find helpful as well. And there's a bunch of journals that have all kinds of information that might be interesting. So if you have an area of interest to you, you could do quick on one of those. So Larry, that's all I have. Did you want to get started now on your part?
Sure.
All right. And we're going to, you have this on a thumb drive now?
Take a couple of minutes.
Yeah, all right. So you guys could chat between yourselves for a minute while we get this loaded on my computer, okay? And I got to give you all these.
This is on here now, for sure.
All right.
All right, so let's see.
Put this on. This is for the recording.
Okay, all right. Let's see, I'm not sure where this should go. Does it make any difference where this goes?
I think you can put it on your shirt. That'd be great, but you also got to get this on, which you might want to put on your pocket. That's for the room.
Right. Okay, and it's just, and this needs to go like,
Either way.
this needs to go like this? Just in there, okay.
Did you want this clip here, or do you want this clip? This one?
Oh, I'll use theirs. And then you can keep yours.
All right, let's try this again.
It better be here this time. Yep.
This?
Yes, yeah, in there. Okay, and there we are. So there should be something, introductory lecture. There we go.
Got it. Awesome.
Finally there, yeah.
All right.
Okay.
What's that? 1:52. So you have about half an hour. Okay?
Yeah, and I might not take that long today. I may just cut it off. We'll see how it goes. Okay.